Well, hello, everyone. I'm Dr. Terry Constantine, the CEO of Brain Research Foundation. It's my pleasure to introduce Dr. Barbara Waszczak from Northeastern University. Dr. Waszczak was one of our 2018 Opioid Initiative Grant winners, and she's going to tell us about some of her research from this project and how this grant actually took her science in an unexpected direction: opioid abuse relapse research. We will save some time at the end of the talk for questions. Now let me turn it over to Dr. Waszczak. Barbara?
Thank you, Terry. It's wonderful to be here. I am so honored to have been asked to talk about the work that you funded. You got us started in an area of research that was new for me, and I'm forever grateful that I've had this opportunity. I'll talk a little bit about my previous work as I go through my slides tonight, but this work, funded by the Brain Research Foundation, was to look for a treatment that could reduce relapse in patients who are suffering from opioid use disorder, a disorder that is very common, as you all know, in today's society. Let me start with the definition of the problem. First of all, despite the fact that we're in the midst of an opioid epidemic, there are no currently available drugs to treat patients in recovery. You're probably all aware that there are opioid-related drugs that are used in medication-assisted therapy. These are drugs like methadone and buprenorphine that basically maintain the patient in a comfortable state where they're not suffering from symptoms of the disorder, but they're not free of the addictive effects of the drug. So they remain literally tied or attached to an opioid type of drug. If they do undergo withdrawal, and they're able to do so, then the period of abstinence that they enter, they're no longer suffering acute symptoms, but that abstinence is difficult to maintain. It's partly because there is a persistence of what we term drug craving, especially made worse when the patient is exposed to a stressful situation, or they are around individuals or in the places where they've previously used a drug. So abstinence is difficult to maintain, and what ends up happening for almost all patients at some point is that they relapse and they start using drugs again. They may succeed in stopping, but relapse remains a very common problem that all patients who have experienced this disorder continue to suffer. So we would like to devise a treatment that can reduce that craving long-term and lower the risk of the patient wanting to resume use of the drug, even when they're in a situation where they might otherwise be tempted. So we need to develop a way to reduce craving. How might we do that? Well, neuroscientists believe that a treatment that will work in this way is going to have to correct the changes that happen in the brain when a person becomes addicted. And there are lots of hypotheses about how this happens. A major one is that the key change might be that the dopamine reward centers in the brain are involved in a change that makes it difficult to experience pleasure without the use of the drug. And especially what we think happens is that there may be a deficiency in the brain's dopamine pleasure centers, and that this deficiency in the reward system leads to a deficiency in pleasure responses, your ability to feel the rewarding or positive effects of anything that's pleasurable. So patients with this problem have a persistent dysphoria. They just can't feel good about things. They have a hard time experiencing pleasure, and they become impulsive. And that we term a reward deficit. It makes the patient feel really very poorly. And so drug craving is a result of that. It's a part of the reward deficit. And it, as I mentioned before, leads to relapse very commonly. So let's look at this in another way. This is a diagram that I took from a National Institute of Drug Abuse website that shows these dopamine reward neurons in a cut through the center of a human brain. So you're looking sort of down the middle of the brain as if we're cut right down the center. About midway back are these neurons that we term ventral tegmental area dopamine neurons. And they send their fibers forward in the brain to what we call the pleasure centers, the nucleus accumbens and the prefrontal cortex. So when a person takes an opioid drug, it activates these reward neurons. And so I'm going to term that kind of a green effect. And it stimulates these neurons to fire and release dopamine in these reward centers. And that feels good. And anything that feels good is said to be reinforcing. It makes you want to do that thing again. So if it's been a drug, you'll want to take that drug another time. And people do that frequently. They become users of the drug, and eventually they find out that after repeated use, they no longer feel the same level of reward or pleasant feelings. The euphoria is a little less. And so that tempts them to take more of the drug, continue to use it, maybe even use it in higher doses because tolerance develops to it. And essentially what happens is these dopamine neurons become deficient in dopamine. You're working them so hard that they can't keep up with demand. And so you eventually begin to feel a loss of that pleasant response, making you feel you need to take more and more of the drug. So this is the reward deficit, and it causes this craving and the tendency to relapse to drug use. When that occurs, you're really not a happy person, and you really would like to be free of this connection you have to this drug, but it's difficult to get there. It's difficult to go into withdrawal and abstinence. So it's a very difficult illness to suffer from. It's something that is hard to escape once you've gotten to this point. The withdrawal symptoms that occur when you abruptly stop taking the drug are an index of this dopamine deficit. So how did I get here? How did I start being interested in these dopamine neurons and the reward deficit? Well, it was a short trip from my previous work on Parkinson's disease. What we ultimately want to do in both Parkinson's disease and in substance use disorders like opioid use disorder is to restore the dopamine that is lacking due to the disease. In Parkinson's disease, which I have been working in for most of my career, 30 years of research, a different population of dopamine neurons begins to die over the course of many years. The neurons that are very close to the ventral tegmental area neurons die off. They normally release a great deal of dopamine in an adjacent part of the brain called the corpus striatum. And when these cells die, as they do over a period of years, dopamine becomes deficient in that area of the brain that I just mentioned, the striatum. And the symptoms of the disease begin when there's about a 75% loss of dopamine cells. You're probably familiar with the symptoms. I won't go into this in detail, but Parkinson's disease is a movement disorder. People become unable to move with speed. They have bradykinesia or slowness of movement, tremor, and rigidity. It's a very serious disorder. It's progressive. And one of the forms of treatment that people have considered and that I'm working with is to try to restore the dopamine that is lacking in the brain by saving the remaining dopamine neurons. This would be the treatment that I'm proposing also to use for opioid use disorder, but it would be in order to save the remaining sick and dying dopamine neurons in the brain of someone with Parkinson's disease. If we could save the remaining 25%, we might be able to stop Parkinson's disease progression. So thinking about that, I thought, well, there's another disorder where dopamine is thought to be deficient, and that's substance use disorders. In addictive disorders, we think that adjacent population of dopamine neurons in the ventral tegmental area become depleted, exhausted, if you will, because of chronic excessive use of this pathway to produce the rewarding effects of the drug. So I've drawn that in on the same diagram, but this time I'm showing in the green here that these neurons are more centrally located right in the middle part between the two substantia nigris. Their cell bodies are located there, and they send their fibers forward in the brain to a more medial part of the brain where the nucleus accumbens is, and the prefrontal cortex as well, but that area is not shown on this view of the brain. In any event, when the cells become exhausted, they lose their ability to keep up with demand, and dopamine in this pathway becomes depleted. So the idea would be the dopamine deficiency creates the reward deficit and the drug craving that drives relapse. If we could restore the deficient dopamine, we might stop that craving and reduce the likelihood of the person undergoing relapse. Okay, so what can we do to bring that restoration of dopamine back? There is a protein that has been known since the early 1990s that is a particular aid to dopamine neurons, especially dopamine neurons that are undergoing stress or are subject to whatever factors are causing cell death in Parkinson's disease, and in areas where we have dopamine deficiency due to overuse of these neurons. The protein is called glial cell line derived neurotrophic factor. It's made naturally in the brain. It's downregulated in adulthood, more common in early life, and it aids in the recovery of damaged or depleted dopamine neurons. People consider it what we call a trophic factor for dopamine cells. It's been promoted as a treatment for addiction and for Parkinson's disease, and that's why I was studying it initially. But the problem we've all encountered in our studies is that it does not cross the blood-brain barrier. So you can't give it systemically like an injection or as a pill. It does not get into the brain by these normal routes of administration. So I show you that here. It can't get past the so-called blood-brain barrier, which excludes many drugs from entering the brain, and certainly proteins and other types of therapeutics. The thing that we have done that I think brings us to a whole new place in addressing these disorders and the use of GDNF is we've designed an intranasal delivery system that bypasses the blood-brain barrier, and we give the drug intranasally, and it travels from the nasal cavity through fine openings within the upper nasal cavity that lead to the brain, and it introduces the drug, or in this case, the GDNF, to the brain so that it circulates within the brain in the fluid that surrounds the brain structures outside of blood vessels. It doesn't go into the blood. It basically travels outside blood vessels, and it travels very quickly. It gets to the rear of the brain within an hour of intranasal delivery. But we've decided not to give GDNF by this route, but to give the gene that makes GDNF in the brain. And that's a short piece of DNA. And we've got a company we work with, Copernicus Therapeutics, that engineers this GDNF DNA and compacts it into nanoparticles, which otherwise would not cross the blood-brain barrier. But we've shown that we can get the gene nanoparticles into the brain by intranasal delivery. So we've done this in rats or rodents. We've been able to show that we can increase the production of GDNF in the brain initially within days, and for weeks after that, given a single intranasal dose. The treatment does not require surgery. So one of the problems that we've seen with the Parkinson's treatment is that they've used it in clinical trials, but they've had to inject it into the brain through cannulas. This is not surgical. It doesn't require surgical procedure, which basically eliminates most patients from being eligible for this form of treatment. It is non-toxic, and it could be repeated if it's needed. So you could give an additional dose if it's necessary to increase GDNF for a longer period of time. It would be useful both for Parkinson's disease and for substance use disorder. So our hypothesis is that this gene therapy for GDNF given intranasally will be able to correct the dopamine deficiency in the dopamine reward neurons. it will reduce the deficit that causes drug craving, and it will reduce the likelihood of relapse. I like to think of this as a form of treatment that a person might take if they have been a user of drugs for a long period of time. They're tired of being a victim of their abuse of the drug, their addiction. They want it to stop. They've tried to become abstinent. They've succeeded in becoming abstinent, but they're always afraid that relapse will occur, that they'll be tempted to go back to use of the drug. This could be a real aid to people in that situation, especially with counseling, to let them know that this treatment is not an opioid. It will let you become capable of feeling pleasure and normal reward that you have not been able to achieve while you've been addicted to a drug. And it will make it possible for you to resume being so-called normal in the state you were before you became subject to this drug addiction. And that would help people to realize that it's possible to become what they were again before they started the use of the drug, and give them the confidence to resist relapse. It's a novel approach. I think it has great potential as a therapy because it's not invasive, it's not another opioid, and it is, again, repeatable. It's easy to administer. I do think it would be something you would want to have done clinically, not in the hands of the patient themselves, but something that they would go to a visit with their counselor or clinician and could receive the intranasal dose on a treatment schedule that's appropriate for that patient. Now, how did we study this in our lab? What did we do with the grant money that we got from the Brain Research Foundation? Well, we were fortunate to be able to test this prediction in a model of opioid addiction called conditioned place preference. It uses rodents, and basically what it does is conditions the animal to prefer the place in a chamber where he experienced the positive effects of the opioid drug oxycodone. And so I've drawn a little diagram of the chamber here. In fact, this is a picture of the chamber we used. It has two sides, and the walls and the floor and the two sides are different. One has stripes in this case, the other has checks, and the floor pattern is different and feels different to the rat. So what we do is we take a rat, and actually each rat in the study gets the same treatment. On one day, we inject the rat with saline, and then we put him into this chamber for half an hour, and he stays in that chamber until, the end of that period of monitoring. On the alternate days, we take the same animal, we inject them with oxycodone. So they obviously are going to feel the effects of this drug, and we immediately place him into the other chamber so that he begins to associate how he feels when the oxycodone is active and where he feels that way. And we do this alternatively for 8 days. One day they get oxycodone, the opposite day they get saline. And so there are basically 8 days of treatment or conditioning, it's called. And then after that 8th day, they are kept without any access to the drug for a period of at least a week. We use 2 weeks in this study. And then we take them back and put them into the chamber. But in this case, we remove the center door. There's a door that separates these two, so they haven't been able to go back and forth until this day when we do the first, what we call conditioned place preference test. We remove the center door and just put him in the middle and see where he goes. And without a doubt, the animal wants to spend the time in the chamber where he felt good after he got the dose of oxycodone. So we keep track for a period of time, how long do they spend in the oxycodone paired side versus the saline paired side. And the preference for the oxycodone paired side is called conditioned place preference. Okay, after 2 weeks of testing the animal for conditioned place preference, we gave half of them an intranasal dose of saline, and the other half got an intranasal dose of GDNF nanoparticles. So we're testing our treatment at this point. Then we wait another several weeks and we take the animals and we put them back into the chamber with the center door up so they can travel between the two. The saline treated animals still want to go to the chamber where they receive the oxycodone initially. They still prefer that side, but the animals that got GDNF nanoparticles no longer prefer that side. They've lost their preference for the drug paired side. So we think that's an index of a decrease in craving. Then we took the animals and waited another week and tempted them by giving them a low dose of oxycodone before putting them back into the chamber with the door up. The ones that were initially given saline really then preferred the oxycodone paired side. You've given them a little touch of the drug that they were familiar with, and they still remember. They feel the effect of that drug again, and it makes them really want to spend their time in the oxycodone paired side. But the animals that we'd given the GDNF nanoparticles to no longer cared about being the oxycodone paired side. They traveled between both sides. There was no preference for the oxycodone paired side. So this is support for our hypothesis that intranasal delivery of these GDNF nanoparticles can decrease craving and reduce relapse potential. It's a strong support for our hypothesis, and it gave us the impetus to write more grant proposals. And hopefully we are going to continue this work with new funding. We did already do some pilot studies, and we've begun to use a new model that's considered more definitive as a test for reducing craving. And that is what's termed an intravenous self-administration study, also in rodents. And in this study, which I'm not going to show you data from or talk at great length about, the animals have access to a lever in their cage where they can press the lever to receive an intravenous dose of the same drug, oxycodone. And the amount of lever pressing is an index of how much they want the drug. So we've already done a study where we've looked to see whether if they have continuous access for several hours to pressing the lever, they become addicted to the oxycodone. And then if you take them away from the chamber where they receive the drug, put them back into the chamber without access to the drug, they can still press the lever, but they won't get a drug injection. That is the point where you see an increase in lever pressing to a great extent. They keep wanting to press the lever to get more drug. And we've shown that by giving intranasal GDNF nanoparticles, we reduce that lever pressing. The animals no longer press the lever to get oxycodone. And it's only a pilot study. We need to repeat the work, and we hope to do that with more funding. We also want to test whether the other side of the hypothesis is correct. If we do reduce craving and relapse, lever pressing in this model, do we also at the same time reduce the dopamine deficit in those same animals? So we're taking some of these animals and we're going to do a dopamine release study after we sacrifice the animal and remove the brain. So we'll be able to tell whether there was in fact a dopamine deficit and whether there was in fact a restoration of dopamine in the animals that got the GDNF nanoparticles. And then the last thing that we need to do is to determine if we can get the same increase in GDNF in the brain of larger animals, equivalent to humans. So we hope to study intranasal GDNF nanoparticles administered to non-human primates. And if we can get at least a 2 to 3-fold increase in brain levels of GDNF after this treatment, we will have succeeded in showing that it does have potential as a therapeutic approach for patients with opioid use disorder. Positive results in these 3 studies would really go a great distance toward encouraging big pharma companies to invest in us and to commercially develop this approach as a way to treat relapse in patients with opioid use disorder. So I can't thank enough the Brain Research Foundation for getting us started, for giving us the first, money, and therefore the first interest in us and belief in us that this idea had merit. And it changed my research and my professional life completely, that I now have a new approach to science and a new disease situation than I had before. I'm forever grateful for that. So that's the end of my presentation. And if anyone has questions, I'm happy to answer them.
Are there any questions? If there aren't from the people that are watching, I have a couple. One, you mentioned that after repeated use of opioids, right, the reward pathway is altered and it causes cravings.
Mhm.
Is there something different with heroin, or do I not understand? I thought that many people get kind of addicted after one use. What is the difference between heroin and perhaps another opioid?
I don't think
Is there-
heroin's not different, I
Okay.
don't believe, than other opioids. But all that drug, as well as fentanyl and other ones that people might take and become addicted to, all stimulate opioid receptors in the brain, and that's how they produce their effect. Some are more potent than others, but heroin's not different than the rest. They all stimulate these opioid receptors that are responsible for reward. What you might be referring to, Terry, is that sometimes people say that the first time I experienced this, I was helpless. I could not resist doing it more and more and more. And that's what we call the reinforcing effect of the drug. Not everybody experiences that, but some people will say that they felt that way and they rapidly escalated their use of the drug and became addicted to it very quickly. But one dose does not cause this change in the reward pathway that I just mentioned. It would take repeated use to get to that point.
Okay.
Okay.
And if anybody has a question, you just have to unmute yourself and ask it. I have.
Yep.
Go ahead.
This is Dave Fishburne, and just one quick question. If you were to get funding tomorrow that you are requesting, continue to have success, how long might it be, and how long might it be before this potentially could get to market? Are we talking 1 year, 5 years, 2 years?
Well, it depends on how much funding we get.
Okay, well, you get all the money you need.
Okay, if you get all the money you need, and if the studies that I described in that last slide work, meaning we showed that it works in another animal model of opioid disorder, we show that we can get it into the brain and increase GDNF levels in a primate brain by at least 2 to 3-fold. We will probably have pharma companies knocking at our door to invest in us, and that would rapidly increase the speed at which we could get this tested and on the market. Short of that, we are hoping to be able to continue it under funding from the National Institutes of Health, NIDA. That will help us test the hypothesis and take it forward. But the next step after we get through those studies is what we call IND enabling studies, where you have to test the treatment in animals, probably primates, to show that it's safe and effective. And so we would need to be able to get CROs, contract research labs, to take this in, study it under what are called GLP conditions, make sure that it doesn't cause toxicity in the brain or in any other system in that animal, make sure that it does achieve the desired effect. And then we need to scale up production. We need to write an IND application for a new drug. The FDA has to consider that IND application and all the data that we bring to support it and decide that it's worth approving for clinical use. And the IND application, if approved, would let us start a clinical trial. And I think that if funding is sufficient and everything works the way I described, we should be to that point where we can try a clinical study within 5 years.
Wow.
Takes 5 years. to go through all those preliminary studies that demonstrate that it would be safe and that it looks like it has potential. And we're hoping to do that, you know, within the next 5 years. We would know at the end of that period, if we started a clinical study, we would know fairly quickly, I think, whether it has efficacy in patients.
Sure.
And if it did, and it weren't toxic in any way, it was safe, I think the FDA would be very willing to approve this as a treatment. Partly because this is a protein we already make in the brain. It's not an unknown substance or a drug that is not normally present in your brain. And only if they're concerned about the method of administration, the intranasal delivery, or some problem with the nanoparticles, would there be some safety concerns, I think.
Sure.
Right, because aren't there
Thank you.
currently clinical trials looking at GDNF and Parkinson's?
Yes, I should qualify that what I just said about there wouldn't be any safety concerns about GDNF, because yes, you're right, Terry, they have been testing it for Parkinson's disease by injecting it into the brain or putting a cannula into the brain and infusing it. And what they've discovered that could be considered a side effect or a problem is that some of the patients who got it reported weight loss. They lose appetite and they don't eat as much. And weight loss was pretty severe in some of those Parkinson's patients. Interestingly, many of those patients reported that their Parkinson's symptoms were improved, but the potential side effect of weight loss was of concern. And the pharma company that was developing and testing it at that point basically dropped the program. So it's not like it has not succeeded. It's been abandoned. And I hope very much that if we're successful in treating opioid use disorder this way, that Parkinson's will take another look. I'm still very interested in Parkinson's research.
Are there any other questions? All right, I have one final question if there isn't one. Other addictions?
Yes.
Alcohol, food,
Yes.
etc.
Yes. All of those are potentially similar to opioid use disorder, treatable by this method. In fact, the literature that I began reading after I realized that the Parkinson's research funding was drying up, I started looking at the substance use disorder research, and all of these substance use disorders that you described all activate the dopamine reward pathway, all lead to potentially the same kinds of changes, the depletion of dopamine, and all of them could potentially be treated with a strategy like I described. If we could restore the dopamine in those neurons, you might be able to recover from alcohol use disorder, from possibly, I know there's great interest in this, stimulants. Methamphetamine use is very much a problem right now. All of it would have to be tested in separate studies. There'd have to be other, animal studies done to determine that it looks like it has potential. But yes, because they all share the same mechanism of producing addiction and suppressing, dopamine reward circuits, this could be a treatment that would work for many or all of them.
Well, and I'm going to end there because that's a wonderful advertisement for why Brain Research Foundation funds neuroscience and doesn't focus on one certain disease or disorder, because many of them, as we know, are interconnected. And so I want to say thank you so much, Barbara, for this wonderful and interesting talk. I'm so glad that we funded you. And I know that you are as well. And I look forward to hearing about some interesting things that may be on the horizon.